So, dear colleagues, today is our seminar, and we have a guest from ETH Zurich, Professor Bill Morandi. He studied at the same university, ETH Zurich, receiving a bachelor in biology and a master of science in chemical biology. From 2008 to 2012, he pursued his PhD in organic synthesis in the same institution in the labs of Professor Eric Carrera, who, by the way, was also a lecturer of Kharkiv Chemical Seminar. Afterwards, he moved to the California Institute of Technology in Pasadena for a postdoctoral stay with Nobel Prize winner Professor Robert Grubbs. From 2014 to 2018, he was an independent Max Planck Research Group leader at the Max Planck Institute for Kohlenforschung in Germany, before subsequently returning to ETH Zurich in 2018. He is currently full professor of synthetic organic chemistry and heads the Institute of Organic Chemistry at ETH Zurich. His research program targets the design and development of new catalytic reactions for the synthesis of small molecules and materials, as well as the valorization of renewable feedstocks and waste materials. He has received numerous awards, among them a Novartis Early Career Award in Organic Chemistry, the Kai Duisberg Memorial Prize from the German Chemical Society, and the selection as a Chemical Engineering Youth Talented 12. He is currently an associate editor for Organic Chemistry Frontiers, a journal from Royal Society of Chemistry. And Jabil, please, you can start your lecture.
Many thanks, Valentin, for this overly kind introduction. I'm very pleased to join you today virtually to share some of our science with you, and of course, sort of make my small contribution to the Ukrainian chemistry community. So I'd like to tell you a little bit about some recent chemistry from my group. So I have selected some topics. Maybe I'll start a little bit with the outline of the presentation today. So something we've been quite interested in the recent years is developing new methods for molecular editing, and this is something I'll talk about a little bit today. Most notably, those two reactions that I'll discuss a little bit more in detail. This transformation that enables us to introduce just a single nitrogen atom into important starting materials such as indoles to generate sort of interesting derivatives. We've also been interested in using the power of organometallic chemistry to do molecular editing of heterocycles, as shown here also on the slide. Then in the second part of the lecture, I'll change gears a little bit, sort of summarizing some of the recent developments in the area of reversible catalysis, which sort of historically has been a very important research topic in my lab. And towards the end of the presentation, I'll sort of merge the two research areas by showing you a reversible catalytic transformation, but that I think is particularly relevant again in this context or this idea of molecular editing. There are a few other things I will not have time to talk about today, but I think they're quite interesting topics. So if you're interested to look into them a little bit by yourself, I've written the reference on this slide. So for example, we've been interested in sort of new approaches to cross-coupling shown here. So a student of mine, Sven, has developed this very creative radical approach to sort of address many challenges in aliphatic cross-coupling reactions. We've also been interested in sort of developing new concepts in cross-coupling with regards to what you can do with the type of electrophiles that you use. And I had another great student who has left a few months ago who has developed this idea of non-innocent electrophiles that can really enable you to do interesting sort of variations of cross-coupling reactions. And we also still heavily involved in sort of fundamental organometallic chemistry, particularly with regards to synthesizing well-defined nickel-1 complexes and study their chemistry, their reactivity in stoichiometric and catalytic reactions. But as I said, for today, I'll focus on those 5 main topics that I show here on this slide. So a little bit of context before I start on this single nitrogen atom chemistry that I've talked about. A little bit of context about our program on amination reactions, amination catalysis. This is something we've been working on also for many years. Of course, there is a broad interest worldwide, many groups working in this area, but in our group, we've been particularly interested in sort of utilizing this reagent shown here, this very simple hydroxylamine derivative. And so what we have done over the years with this reagent, just very summarized fashion here on the slide, we've been interested in sort of inserting or installing free NH2 groups in all sorts of hydrocarbon starting materials, with the idea of being able to rapidly synthesize sort of important amine containing building blocks. And here on this slide, you see a summary of those efforts that took a few years to develop. So as I said, we've been using this reagent shown here in combination with iron as a very efficient and very sustainable type of catalyst or metal for those transformations. We've been able, for example, to do CH amination chemistry, directly introducing the unprotected NH2 functionality. We've also been able to do all sorts of amino functionalization reactions. It goes from amino chlorination, amino hydroxylation, and most recently, amino azidation, which is a quite synthetically useful transformation in the context of accessing sort of diverse diaminated compounds in synthesis. I've also been involved in trying to transfer the amino group onto heteroatoms, for example, sulfur. So we've developed, as shown here, this direct synthesis of unprotected sulfonamides directly starting from simple thioethers. But as I said, this is sort of how we used to do chemistry in the group until sort of 2021. And this is at the time where I had a really, really brilliant PhD student, Julia, that then has now left the group. She's a postdoc at Caltech, and she was interested in doing amination chemistry, but she wasn't really interested in doing sort of the typical alkene amination chemistry that we were focusing on in the group. This is also, for context, right around the time, 2021, where particularly Mark Levin and Sarpong started to really popularize this idea of skeletal editing, or how can you really think about sort of changing the core structure of organic molecules to generate derivatives, which is particularly useful for late-stage functionalization. And sort of, being exposed to this and being interested by the amination chemistry going on in the group, Julia came up with this idea shown here, at least this dream reaction that she wanted to develop. That would be about doing an amination reaction, but instead of doing an amination that would do, for example, a CH functionalization on the periphery of the molecule, she was really interested in trying to develop the reaction shown here that would, mean having direct insertion of just a single nitrogen atom into the core skeleton of an indole starting material. Fundamentally interesting type of reactions, but also if you think in terms of potential applications, very powerful, because you obviously all know that, indoles are widespread in, a widespread motif in all sorts of bioactive compounds, right? Drugs and natural products. So that would be a very powerful way to diversify the indole skeleton in those bioactive compounds and sort of generate an analogous heterocycle, and that could be very interesting for drug development. So the idea how to develop this reaction, again, this credit goes to Julia, was to develop a reaction that would be analogous to a Chamichian-Denstedt reaction, which is sort of the exact same transformation I'm showing here in the middle, but using a carbene instead of a nitrene type intermediates. And so inspired by this classical name reaction, Julia thought that if she could identify conditions to generate a highly reactive nitrene type intermediate that would have a good leaving group on the nitrogen, potentially she could access this type of intermediates here, those aziridines or aziridiniums, that hopefully, because of their instability, because of the good leaving group ability of the phenyl iodide here on the nitrogen, would trigger a fragmentation or re-expansion process that would directly lead to the formation of the quinazoline product of this transformation. So Julia started to work on this project. I mean, I should say she was joined by a really great team of other people in the group, in particular Ori, here was very involved as well in developing this powerful transformation. So where did it all start? I mean, obviously it started with trying to find the good conditions, trying to optimize the process. And there are a few features that I think are quite interesting, or sort of take-home lessons from this slide shown here, this sort of preliminary optimization. We were originally trying quite a lot to get free NH indoles to work in this chemistry, but at least back then this was something that was very challenging. And so the key was really at the time to identify sort of a suitable protecting, or I should say steering group on the nitrogen that would sort of enable this reactivity, this transformation. So silicon ended up being really, really good at enabling this chemistry. I mean, we think for two main reasons that are sort of schematically summarized here on the slide. I mean, on one hand, of course, when the indole is going to react with that reactive nitrine species, it's going to play the role of a nucleophile. So you do want to avoid having, a decrease of the electron density of your indole starting material. And obviously having a silicon substituent on the nitrogen versus having, for example, a Bach protecting group, of course, is very helpful in this context. At the same time, at the end of the reaction, you want to have your silicon protecting group potentially being removed under the mild reaction conditions to directly release the actual quinazoline product you want to form in those transformation. And silicon is of course well known as a really good electrofuge, particularly under conditions where you have methanol and water around. You can very easily attack the silicon and leave the two electrons on the product and directly generate the product of the transformation. So that was kind of a key design element in getting this reaction to work, and subsequently, of course, just further optimizing the reaction, playing a little bit with the amount of oxidant and the amount of ammonia or ammonia source under those reaction conditions could have a very high yield of this transformation. You see here a picture. So this is a transformation that is extremely easy to set up. So it's like an open flask reaction. You dissolve your substrate in methanol, you cool down in an ice bath, and then you add the two active reagents, and you get the reaction done in a matter of a few minutes. It's a very, very fast reaction. So actually very convenient process. So what can you do with this transformation? Obviously, one sort of obvious thing we wanted to probe was whether substituents at the 2 and 3 position of the indole would be tolerated, because most of the bioactive indole scaffolds or substrates have substituents at either of those positions or even both. You can see here in this first entry on this slide that the reaction, even having the disubstituted system or substrate, was working really well with high efficiency for the introduction of the nitrogen at this position. And next that kind of raised the question of what would happen if we start to tether those two substituents together in a ring system. Because you could imagine that as the ring gets smaller, potentially generating the desired quinazoline of the quinazoline product of the transformation would lead to the generation of highly strained bicyclic systems. And so we're really interested to probe this, and you can see that when you have a seven-membered ring as a substrate, you sort of reach the limit of this reaction, in the sense that you still get insertion of the nitrogen at the correct position, but obviously due to the high strained nature of the product of this reaction, it doesn't fully rearomatize to make the quinazoline, and rather stays at the sort of methanol adduct state of the product. That made us very curious to see what happens if we keep reducing the ring size in our polycyclic indole starting materials. You can see that when we go to 6 and 5-membered ring, now energetically it's very challenging to access the desired quinazoline product. And so the system finds an alternative rearrangement pathway to generate another stable heterocycle in this process with very, very high efficiency, which is obviously quite interesting example of sort of substrate control chemodivergence. What is happening in this reaction? We haven't done yet any careful mechanistic studies, but we think also based on some preliminary DFT calculations that we first have a common intermediate in both cases. So we have this adduct that results from the attack of the nucleophilic indole C3 position onto this proposed iodonitrine intermediate. If we go through the normal pathway on the left, we have a ring closure here to generate the aziridine. So this is just effectively a stepwise aziridination process. And then we have this fragmentation that generate the sort of normal product of the transformation, the quinazoline. You can see, of course, the primary product of this reaction is still the silyl protected sort of species, but this is obviously very readily, we think, hydrolyzed under the reaction conditions, in the presence of methanol and traces of water. Perhaps more interesting to look at the sort of unexpected pathway to the quinoxal. Telling you, right, this is very interesting transformation, and obviously we're interested in terms of synthesis, right, to apply this transformation. I should say that, in terms of functional group tolerance, that's a very pleasant reaction to work with. I mean, you can see here just sort of 5 examples. Of course, we have many more entries in the substrate scope of this transformation, but we've been able to apply this reaction to quite a few starting materials where we have unprotected functional groups, right? Things like carboxylic acids, amines, and many other things. And so that makes this transformation, we think, quite useful in terms of synthetic application, because now you can really take sort of complex advanced intermediate or even already existing bioactive compounds, and you're able in sort of a one-step process to change the nature, the core of this heterocyclic part of those molecules to a different one. And we think particularly for medicinal chemistry, this is something. Right, let's continue with molecular editing. So we have now, as I told you, a little bit of single nitrogen atom insertion. And we've also been interested in taking sort of a completely different approach to this idea, to this concept, leveraging sort of organometallic chemistry. A little bit of context maybe about this transformation. So this is, I think, something you all know well. We have, you I think all familiar with this type of chemistry. It's classical metal halogen exchange reactions. Very powerful tool where you can take sort of any aryl halides. Typically, you can turn it into an organometallic reagent, and then you can do a lot of very interesting and useful chemistry to this type of Umpolo reactivity. And so we've been very interested, particularly with my postdoc, Hongyu, I'll show a picture of him in a second, in trying to extend a little bit this idea or this concept to actually finding ways to exchange core atoms like a carbon that are present in the ring system, also with a metal species, to try to access those cyclometallated intermediate or reagents, and then use this as sort of a platform to diversify, what we can do with those intermediates and those starting materials to generate interesting sort of cyclic structures, right? Sort of starting in all cases from one common starting material, but taking advantage of the interesting reactivity of the metal, being able to access many sort of interesting derivatives. Again, with this sort of idea of molecular editing in mind. In terms of design, we looked at lactams to develop this chemistry. Of course, lactams are very common, also in bioactive structures, so it's fundamentally interesting to do chemistry with them. And based on literature showing that nickel zero can be quite active towards oxidative addition for this type of functional group, we thought that if we would apply this logic to lactams, we might be able to have clean oxidative addition followed by decarbonylation to generate this intermediate here. And that would be sort of an in situ, through this decarbonylative process, an exchange of that CO unit in that starting material for indeed a metal fragment, in that case, this nickel fragment, that then we could use to sort of derivatize those structures in many, many interesting fashions. So this chemistry started really as an organometallic project. So we were really interested first to probe this reactivity in an organometallic fashion. So we looked at many different lactams, many different ring sizes, many different types of hybridization, so both sp2 and sp3 system, really trying to show the generality of this type of organometallic reaction. And, this is a very sort of complete publication. So, if you're more of an organometallic chemist, I think it's very interesting to look a little bit at the details, because for most of those reactions, most of those systems, we actually got crystal structures and very careful characterization of all the products. But the point, again, sort of here just to show that, this is very general, this sort of oxidative addition decarbonylation sequence. In terms of organometallic chemistry, it was also a highlight because we could show, I think it was the first time, if I recall correctly, that we would actually isolate and crystallographically characterize a sort of well-defined acyl nickel complex that is the oxidative addition complex from this reaction between a well-defined nickel zero species and an amide bond. I mean, in that case, a lactam. And so there was a sort of a trophy molecule that was very interested in terms of organometallic chemistry to sort of characterize, to sort of identify. Anyway, you're probably more interested in terms of what we can use this chemistry for in terms of organic chemistry of synthetic applications. So it's still a stoichiometric stepwise process, but we can do a lot of very interesting chemistry. For example, we can generate acetidines by triggering the reductive elimination of this carbon-nitrogen bond by oxidizing the nickel, first from a nickel 2 to a nickel 3 species. If you have an sp2 hybridized system, you can actually thermally trigger the reductive elimination to generate the corresponding product. So this is possible in those cases. But something we were, of course, really interested in terms of showing a real direction on how you could apply this type of chemistry in the context of molecular editing was to take this quite important class of bioactive molecules, those azasteroids, show that we could very cleanly react at this position here, do the oxidative addition decarbonylation, and then from that intermediate here trigger two interesting sort of downstream transformations. One of them is, again, by first oxidizing the nickel in situ, we can trigger a very clean reductive elimination. You can see here we have retention of all the stereochemical information that we have in the starting material. So quite clean process. And something else we could also already show with this chemistry is that we can add again CO to those organometallic species, and that can be used to actually trigger the reverse process. So reinsert a molecule of CO, do the reductive elimination. Obviously, that could sound completely useless because you're going back to the same structure where you started this transformation. But of course, this can be very useful in the context of isotopically labeling your molecule, because now you can start with a C12 CO natural product or drug derivative, and now you have the ability to get back in with the label source of CO. And so this is also something now that we're doing a little bit more in collaboration with Professor Shibli at ETH, even with regards to doing radio labeling with this transformation, because this reaction is actually extremely fast. I mean, here it says 16 hours, but it's, of course, much, much faster than this. All right, so those were two examples of sort of molecular editing. Now I'll tell you a little bit more about reversible catalysis. For this, I think it's very useful to refer to the very first reaction I talked about today, I think, as a platform to introduce this section of the presentation. So this is, again, sort of a summarized version of the transformation I talked to you about in the first part of the presentation, this indole to quinazoline ring expansion that I think nicely illustrates how we often do chemistry, how we often develop organic transformation, because we really, in that case, rely, we think, on those highly reactive, hypervalent iodine reagents in this transformation. And this is kind of, as I mentioned, very typical in our strategy in organic chemistry, where many people have developed extremely useful and powerful reactions that use sort of the high instability or high reactivity of all those interesting classes of reagents, right? Hypervalent iodine reagent, diazo compounds, azides, strong oxidants, strong reducing agents. And really the overall strategy is to use sort of or leverage the instability of those reagents, the fact that they're high in energy, to of course trigger the desired functionalization or defunctionalization reaction that you want to do in terms of organic synthesis. And this is, of course, nothing fundamentally wrong. I mean, you've seen obviously in the first project today, we still do a lot of this chemistry in MATLAB. But in some cases, it can lead to challenges because obviously those reagents, because they're highly unstable, because they're highly reactive, they also tend to be not the most convenient reagents to use in the lab, right? They tend to be explosive. They also tend to be highly reactive towards biological systems. So they tend to be very toxic, for example. And so we've been kind of interested to sort of think about new or alternative approaches to sort of do those kind of functional group manipulations in organic synthesis without having to rely on using those reagents, because we thought that could be a very interesting sort of new angle on functional group interconversions. The inspiration on how to develop this kind of transformations came from transfer hydrogenation. This is a very powerful, very well-known transformation where a catalyst allows you to reversibly transfer an H2 molecule between two stable starting materials, right? A carbonyl and a secondary alcohol. This is effectively two reactions for sort of the price or the cost of developing one, because if you play a little bit with the position of the equilibrium in those transformations, you can actually push the reaction forward. So have a very general strategy for reducing ketone just by using isopropanol as a solvent. This is a way to push the equilibrium to the right-hand side of the equation, and then you have this very general reduction process. And you can use the same exact conditions in reverse just by having acetone as a solvent in excess as an oxidant. And now you can sort of push the equilibrium in the reverse direction, if you wish, to have a very general oxidation process. And so effectively what that means is that this gives you one set of conditions, one catalyst effectively, but you can do two different very useful transformations with this. So this is sort of one lesson you can learn from this transfer hydrogenation. Another lesson, another feature that's very interesting is it allows you to do reductions in this direction, in this sense, without having to use hydrogen gas as a reagent. Particularly for small lab scale reactions, this can actually be a very convenient sort of alternative to the normal approach. So inspired by this sort of classes of reactions, transfer hydrogenation, we became very interested in trying to extend this concept to transfer reactions that go beyond just transfer hydrogenation. And this is, again, just transfer hydrogenation in sort of a very schematic way. And our idea was, can we sort of, again, leverage this concept and develop reactions where now we're transferring actual functional groups, actual functionalities between two stable starting materials. Those would also be reversible reactions, so it's very analogous to transfer hydrogenation. And, again, we would also here have to think about ways how to control the position of the equilibrium with suitable driving forces, right? In many cases, just using Le Chatelier's principle. So, if one of the side product is a gas, or if you use something in excess, or if something precipitates, you have ways to drive the reaction in one direction, and then you can, probably use the same catalytic reaction in reverse and just find another driving force. And of course, an essential sort of feature of this approach is that many of the things we could transfer here between two stable starting material would hopefully be things that are extremely reactive and sort of, not convenient to use on their own as sort of, well-defined reagents. Anyway, to keep this short, I'll just show you now first one very short summary of the very first reaction we developed back in 2016. Using this principle, there was a transfer hydrocyanation using a nickel catalytic system in the presence of a Lewis acid. The idea, again, is very simple. If you look at this equation here at the top, you see that you're transferring formerly HCN from a rather stable aliphatic nitrile onto an accepting alkene to generate a hydrocyanated product, while at the same time you do retrohydrocyanation, of course, of your reagent to generate, the corresponding alkene. What we were really excited to show back then was that we could have full control over the direction of this reversible reaction. So if we want to have a very general hydrocyanation procedure, we just use this small, low molecular weight alkyl nitrile. So we generate the gas as a byproduct. The gas can escape, and this drives the reaction. In contrast, if we want to reverse the reaction and push it backwards, now we can have ring strain release in the hydrocyanation process, sort of in the accepting process. And this provides a very powerful driving force to push the reaction now in the reverse direction. And we have a very general way to perform retrohydrocyanations to generate the corresponding alkene. And so this type of reactions, we've developed many in the group since 2016. I, of course, don't have time to show all of them today, but they generally rely on this sort of simple mechanistic principle here. So you need to have a low valence transition metal, for example, nickel in the hydrocyanation, that can do oxidative addition into this carbon functional group bonds. Imagine the functional group is the cyanide, right? If you want to relate to the previous slide, you go to this intermediate here, then it's just simple organometallic chemistry. It's beta hydride elimination. You do a ligand exchange process with your alkene, and then you start to go back in reverse. So through the same steps, but just in reverse direction. So you do a migratory insertion into your metal hydride and a final reductive elimination. So something we were interested to do for years was to use this principle for difunctionalization. So instead of transferring one H and one X group, can we actually think about transferring two different functional groups, or at least two times the same functional group? Using the same principle, this is very difficult if you look at this very, again, simple mechanistic rationale, because even if we're able here to control the selectivity of the beta X elimination, because now we would need to eliminate this X group over the H group. So this is already the first challenge in those reactions. But even if we're able to overcome this challenge, we would still have an issue doing this reinsertion step, because thermodynamically, this is a very disfavorable step to insert an alkene into those sort of metal halide bonds, for example. Anyway, we've been interested now over the years to start to study this a little bit more systematically from sort of an organometallic perspective. I don't have time to give you the details of those mechanistic studies today, but if you're interested, Michael and Oliveira and my group have studied very carefully this step here, the selectivity factors that sort of control whether a beta X group or a beta H group gets eliminated from the corresponding palladium alkyl intermediate. So I really encourage you to look at this publication if you're interested by this. Anyway, we're a synthetic organic group. And so, obviously, besides doing those organometallic studies, we're still being very interested to develop those reactions, using any other approaches we could think of. And that took quite a few years, and it sort of the success resulted from a very nice collaboration with a group of professors, Siegfried Waldvogel, shown here on this slide, who's really one of the world experts in sort of applying electrochemistry to organic synthesis. And so I had a visiting PhD student from Siggy's group back in 2020 for, I think, 6 months. This guy, Johannes, shown here. We started to collaborate with a good postdoc, really excellent postdoc I had at the time, Shishang, and they came up with this sort of hypothesis that probably trying to address this challenge of difunctionalization with electrochemistry would be sort of the logical step to be successful in developing this reaction. The rationale behind this is actually very, very simple. So I showed you on the previous slide, if I go back, that one of the big issue in doing difunctionalization is the selectivity here, right? If you have an alkyl transition metal species, doing beta hydride elimination is likely to be as easy, if not easier, than the beta X elimination, or at least it's really not clear which one will necessarily eliminate. If instead you think about doing an electrochemical approach that proceeds through single electron transfer steps, this is shown here, and as I said, this is quite a simple rationale. You would eventually, after two sequential reduction process, arrive at this carbon ion intermediate shown here. That obviously then would have no other choice than eliminate an X- group, because a hydride is obviously not a good leaving group in organic chemistry. So this very simple rationale we thought should effectively help us to get the elimination, the very selective elimination of the X- over the H-, and sort of drive the reaction in the direction we want. Another benefit of sort of merging this idea of transfer reactions with electrochemistry is that it leads to an extremely simple cell design, because if you think about it, in those transfer reaction we developed in our group, effectively those transformations are redox neutral, and also they're atom neutral, right? You don't have consumption or release of any atoms or electrons in those processes. And that allows you to have very, very extremely simple cell design, shown here, because you do not need to couple your transformation with any sacrificial process that takes place, for example, at the anode. And so what effectively you can do is pretty much use electrons, if you wish, as mediators, or maybe even as catalysts in this transformation. They will just pass through your reaction mixture, help you decompose your donor, and then you will have reoxidation at the anode. So, all the electrons will go away again. You will start to form all the bonds you need on the accepting molecule, and that's leading to the product of this transformation. So actually leads to a very simple design that we were able in my lab to set up back then, despite not being experts in this area. Anyway, what can we do with this chemistry? Synthetically, we can do transfer dihalogenations, and this is something we tried for years. Using nickel catalysis and palladium catalysis, using sort of the older approach we were focusing on, and we're always completely very, very far from getting any success. And so this electrochemical strategy was very powerful. So we could do transfer dibromination. So now you can think about using something as simple and stable as dibromoethane as a surrogate for Br2. You can dibrominate quite a wide range of different alkene acceptors. Electrochemistry, I learned now over the years with SiGe, is also very scalable, particularly if you have this type of undivided cell with very inexpensive graphite electrodes. You can very readily scale up those transformations. And so that could be sort of an interesting strategy if you have to make a few grams of some dibrominated compound. We can also do transfer dichlorination reaction using this approach. So this also works really well. So, again, you can avoid the use of Cl2, and you can generate dichlorinated products that, are, of course, also quite interesting synthetic intermediates. Now, something I haven't mentioned or I haven't highlighted so far in this part of the talk, in the reversible part of the talk, is that obviously one benefit of developing catalytic reversible reactions is that we can, in theory, think about using the reaction in the forward sense, right, to functionalize a given starting material and maybe make a useful product that can be used for an interesting application. But this is not the only thing, right? Obviously, because the reaction is reversible, you could then think about using the same reaction in reverse to recycle those functional molecules at the end of the life cycle, and potentially use the sort of power of reversibility to develop sort of closed loop applications, where you can use the same reaction to make a functional useful molecule, and then use the same reaction to recycle it at the end of the life cycle of the lifetime, as I mentioned. So one example where we think this is quite relevant, particularly with halogenated molecules, is obviously those are sort of typical structures in persistent organic pollutants. And so, of course, there are many fluorinated structures that are problematic, but also brominated and fluorinated compounds have been used a lot in many, many applications. on huge scales, right? In for example, as agrochemicals, but sometimes also as flame retardants and you know, many other different type of applications. And their persistent nature is really a big problem. And so we started to think that maybe our chemistry in reverse could be very interesting in trying to recycle or upcycle some of those waste materials. In one compound we started to focus quite a lot a few years ago was lindane. So lindane is an example of a persistent organic pollutant. It's an insecticide. It used to be produced on really large scale in the 60s, in the 70s. And the big problem is that in the synthesis of this compound, actually just a little bit of the yield was the actual desirable isomer that could then be used as a pesticide in sort of the desired application. And what companies back then would do with the unreactive, undesired isomers, they would just landfill them directly next to big production sites, to big chemical sites. And this has led to, you know, quite some contamination. And so there's still quite a few of those landfills around the world that are basically a mix of sort of soil, of course, that is around the chemical production side, mixed with really high concentration of those lindane isomers. And so with my postdoc, Shishang, back then, we were really interested to test, at least as a proof of concept, whether we could potentially use our reversible dichlorination process as a way to remove all those chlorine atoms from the lindane, from this waste compounds, and try to transfer them in hopefully a productive fashion onto another alkene acceptor, with the ultimate goal of potentially trying to make new valuable sort of organic intermediates or products starting from those waste materials, right, from those contaminated soil samples. And that was quite an interesting type of transformation, or an interesting project. So Shishang actually collected some soil samples, uncontaminated one, just around our campus, and just mixed them in a one-to-one fashion with pure lindane to sort of mimic, you know, the sort of nature or composition of actual samples from landfills. I mean, you can see here on this slide, on this sort of short table, that it was working remarkably well. So without any pretreatment, so just suspending the actual contaminated soil sample, the reaction was working really well. So really a robust process. And since then, we have been able to actually test this chemistry on some real samples. And this has looked very, very promising. So, you know, this is something that, you know, we don't know for sure whether it's going to lead to a real application, but at least as a proof of principle, we think this is, you know, quite an interesting way of showing that reversible reactions could in principle also lead to recycling or upcycling of waste materials, which is obviously an important topic nowadays. Something else on the sort of reversible catalysis that I'd like a little bit to mention or talk about today is this idea that this shuttle catalysis principle really allows you to synthesize highly reactive species, or at least introduce highly reactive functionalities in starting materials. And this is much easier with this approach, because if you think about it, by design, we need, or we want, our products to be reactive towards our catalyst, because the type or the nature of our products is the same as the reagents that we have in our reactions, right? We really rely on having the donor molecule being always very reactive towards the catalyst, just really enabling this reversible transfer process. And this feature sort of allows you to really leverage this in developing transfer reactions where you can access highly reactive species, again, because of sort of the strategy, or because of this principle. This is not our work, this is from the literature, but I think this beautifully illustrates this idea. So quite a few groups have developed ways to do transfer hydromagnesiation processes. So you can actually take a donor Grignard molecule in the presence of iron and copper, two different catalysts. What you're able to do is to transfer formally this H and the magnesium X onto an accepting alkene to generate new Grignard reagents, you know, for example, in an in situ fashion, and then trap them with all sorts of electrophiles. So quite a remarkable transfer reaction that I think really shows that you can transfer highly reactive functionalities using this strategy, this process. Something from our group to highlight this idea is something we did quite a few years back, now 6 years ago. We developed this transformation that is an alternative to rapid type carbonylation reactions, where for the first time we were able to generate acid chloride products starting from simple alkene starting materials. And again, the principle here is the same. If you try to do a normal type rapid carbonylation reaction, so if you use CO and HCl to generate acid chlorides from alkenes, this is something that's very challenging, because if you think about it, your acid chloride, of course, is a very reactive electrophile towards the palladium center. And so that can actually trigger many side reactions, a lot of downstream reactivity that you would like to avoid. But if you design a transfer process, as I mentioned, by design, you want now your catalytic system, the palladium, to be highly reactive towards the acid chloride, because that's going to be the starting point of your catalytic reaction. The palladium is going to attack the acid chloride. It's going to decompose it to its elements. It's going to transfer to the other alkene and regenerate the same bonds. And this is a reversible process, and we just rely on driving the equilibrium to the direction we want. So I think those two examples really highlight this feature of transferring highly reactive functional groups between molecules. And so sort of following on this sort of short introduction for this idea of why shuttle catalysis is advantageous for this, I want to now tell you about some really recent work from a great postdoc in my group, Tanner, who has been really interested in sort of leveraging this idea to access other interesting type of functionality or transfer them, in a shuttle catalysis process. The type of reaction he has been interested to sort of focus on were those cobalt MHAT type reactions. This is a very well-established research area. Many big contributions originally from Mukuyama, from my colleague Eric here at ETH, from the Shenvi group, and many others. And you can do a lot of very useful hydrofunctionalization chemistry with this. You can generally take unactivated alkenes. You can take all sorts of radical traps. You take a cobalt silane catalyst, usually in those transformation, stoichiometric amount of silane, stoichiometric amount of peroxides in general, and you're able to generate all those functionalized products. If you think about it, and if you look at the literature, there's some limitations in those processes, and one that really got Tanner and my attention was the fact that it was not really possible to do hydroiodination reactions using this chemistry. And again, if you think about it, an alkyl iodide is a quite reactive species, and under those reaction conditions, you can imagine that those products are not really stable, right? And so that really sort of calls for kind of a different approach to develop this chemistry. And so, based on what I just told you on the previous slide, you can imagine that the idea that Tanner had was that if we were to develop a catalytic reversible transfer strategy for this hydroiodination via those cobalt MHAT type mechanisms, we could potentially have really, really mild reaction conditions where, again, by design, we want the iodide to be reactive towards our catalytic system, because, again, we have this continuous sort of reversible transfer process that we will control with, position of the equilibrium. In practice, if you look at it, this would look a little bit like the strategy shown here. So classically, as I showed on the top of this slide, you use stoichiometric oxidant, you use stoichiometric silane. Now, the idea would be to have a transfer hydrofunctionalization strategy. So again, now you would have a molecule instead that would act as the reagent that obviously looks very similar to the product of your transformation. And you would just hope that the catalytic system is able to deconstruct these reagents to transfer the hydrogen and the functional group onto the accepting alkene. So how do you go about this? This is the design that Tanner came up with. So maybe I'll first draw your attention to the sort of normal cobalt HATS catalytic cycle shown here. So I think it's easier to start by looking at those cobalt-3 hydride species. Those are really well known, as I said, based on all the precedents to undergo HATS chemistry with alkenes, even inactivated alkenes, to generate this radical pair here, where you have this carbon-centered radical and the cobalt-2 species. What we were hoping is that this alkyl radical species could intercept a secondary catalytic cycle, a XAT, so an halogen atom transfer catalytic cycle, where it could pick up the functional group, again, maybe to ease following sort of this slide. Think of this think of the iodide here, to pick up formally this iodine dot, from this XAT donor intermediate or molecule, generate the product of the reaction. Then you would have this reduced XAT catalyst that could react with the donor molecule, pick up the functional group, the Fg dot, so the iodide dot, if you wish, generates this secondary radical species that then could recombine with the cobalt to do the elimination processes to generate the corresponding alkene, and that would close both catalytic cycle. I mean, those two catalytic cycles, as I said, actually quite well known. So the cobalt hat has been used in the chemistry I mentioned before, and the area of XAT-based catalysis obviously is something that is very active research topic, and Naguib and Leonori, for example, have made huge contributions in this area. So the idea here was really to find a way to combine the two cycles to try to really enable this transfer strategy to do hydroiodination type reactions. I'll keep this short because I think I should slowly start to move towards a conclusion of my presentation. So this is this idea of transfer hydroiodination. It relies on two catalytic systems. As I mentioned, one catalyst is very typical for this type of transformation is those cobalt silane species. And the co-catalyst to this iodine atom transfer, this XAT transfer, is a chromium-2 species that was identified as the best co-catalyst to do this transformation. And now you can, for the first time, do hydroiodination reactions using this very mild transfer process using those two catalytic species. All right, so those were 2 or 3 examples of sort of shuttle catalysis reaction, how we can have a donor molecule and transfer a group from those stable donor molecules onto an acceptor like an alkene to, transfer the functional group and generate interesting products without having to rely on using highly reactive intermediates like, nitrines, carbenes, HCl, HCN, etc. A few years ago, we became interested in sort of extending this concept to having 2 molecules that can actually each sort of contribute with one functional group to sort of transformation and exchange those 2 functional groups in a transfer process. Just to keep this sort of story short, I'll just summarize here a little bit in terms of concept, how those reactions would look like if you just redraw this equation here by having explicitly drawn the single bond that is between a substrate scaffold and the functional group. You see that those transformations start to look a little bit like metathesis reactions. And obviously, mechanistically, it's going to be very different from alkene metathesis, but you probably see a little bit the analogy, right? You effectively also cutting 2 bonds in this case. They're just 2 single bonds. And you're reversibly sort of rearranging or redistributing all the substituents on those 2 starting materials to, generate sort of crossover products that as I said, in those transformations. And so, if you think about this, you can imagine that those reactions could potentially be interesting to look at in terms of synthesis. This section is more focused a little bit on the type of applications. I will not revisit all the reaction development we have done in this area, but one reaction we have used a lot in different applications is the one shown here. This is what we call a carbon-sulfur bond metathesis reaction. So, of course, in this slide shown here, this is not a particularly useful transformation. This is just a model reaction we use to optimize this process. that allows you to cut reversibly carbon-sulfur bonds and exchange these in a library of two substrates to generate the corresponding hetero product in this transformation. For this, we have to use very electron-rich palladium-0 species that have NHC-based ligands, because those are really, really important to sort of enable the otherwise challenging oxidative addition into unactivated carbon-sulfur bonds. As I said, I'd rather focus a little bit on the application of this chemistry in this short section. So we've applied this reaction in sort of generating libraries of derivative. This is very useful application, we think, of this chemistry, because you can take fairly complex, potentially a biologically active molecule, and now you're able to very efficiently sort of change the nature of the substituents that you have on different aryl thioethers that you have in those substrates that you use as starting materials. But the real reason I'm showing this today is that that was sort of a start in my group in sort of pushing this area, this chemistry, towards trying to make and break materials, in particular polymeric materials. And so the very first application of this is the one shown here that we had in this original publication, where we realized that this polymer, polyphenylene sulfide, it's a 1D thermoplastic that is used as a replacement for metallic parts in many sort of high-level applications. We thought that it would be interesting, again, this idea that our chemistry could be used in reverse to recycle things, to see whether we could completely depolymerize one of those polymeric material to generate sort of the smallest possible cross-coupled monomeric building block, with the idea that potentially you could use this as a way to generate interesting organic building blocks from waste that can be used in interesting synthetic applications. So that was how it sort of started. But then I had really brilliant postdoc. I'll show his picture in a second, Miguel. And he had a background a little bit more in material science. And so together with him, what we really tried to do was to try to harness the reversibility of those CS bond cleavage reaction, not only in cleaving polymeric structures, but also trying to use the reaction in reverse also to make new polymeric structures, because we thought that that could lead to very interesting applications in theory, where we could use this reversible reaction to first make an interesting polymer, use it in some application, and then at the end again of the life cycle, in theory, you could recycle those polymer, or at least depolymerize them back to simple monomeric structures. An area where we thought this would be particularly exciting is sort of summarized here with those two TOC pictures. So you're probably aware that the synthesis of covalent organic frameworks is a very, very active, very important area in organic materials. And the synthesis of those sort of perfect materials really relies on using the self-correcting ability of reversible reactions. And so in general, what reactions people use in this area are sort of the typical reversible organic transformations. One example is shown here, is the synthesis of boronic boron esters, or for example, imine exchange reactions. Those are the very typical fast reversible reactions that people have used to really be able to synthesize those perfect 2 or 3D materials. The problem with this is that those type of bonds are actually quite labile. And so particularly towards hydrolysis or high temperatures or harsh reaction conditions, those materials tend to be relatively unstable compared to what you would like them to be for many applications, right? That might require high temperature or high, really harsh reaction conditions. This really contrasts again with this material that I just showed before, polyphenylene sulfide, because this is formed from very stable carbon-sulfur bonds. This gives very, very interesting physical properties to this 1D polymer. I mean, as I mentioned, it's very often used as a replacement for metallic parts. So, it's extremely resistant to high temperatures, to acids, to water, so hydrolysis, etc. And so now if you remember about what I told you on the previous two slides, we have a way to reversibly cleave carbon-sulfur bonds. So what about designing monomers based on aryl thioethers that could potentially polymerize and really take advantage of the self-correcting ability of our reversible reaction to generate porous polymers that would have this sort of benefit in terms of stability of what you would find in the 1D material that is based on those very, very strong and unstable carbon-sulfur bonds. I realize this is a little bit of a longer introduction, but really kind of to put things in context. And, just unfortunately have to summarize this with sort of one scheme, the really amazing work of Miguel and Yorios on this project. But I think the message is really clear. They were able to design 7 different monomers. They were able to polymerize them to make new porous materials in very high yields. Those porous materials have interesting physical properties. So, they have decently high porosity. I mean, this is still something we're, of course, trying to further improve in the group. But very interestingly, we've done quite a few chemical and physical tests of those materials. And as predicted, they're extremely stable to hydrolysis. They're extremely stable to high temperatures. They're very stable to things such as concentrated sulfuric acid. So they really have those very sort of interesting properties of the 1D material. But ultimately, what we wanted to do or show with this chemistry as a proof of concept is that in theory, we can now make, or in practice, we can really make those new materials that have those interesting physical properties. But as you can see here on this slide, we can actually use the same catalytic process in reverse to completely depolymerize those materials, to completely recycle them. And so I really think this kind of shows this idea of using those reversible reactions, potentially also in the material science, to synthesize new materials with very interesting physical and chemical properties, but still being able to recycle them potentially at the end of the life cycle, after you've used them in some interesting applications. Something else you can do with a reversible reaction that is self-correcting is you can make large rings. This is a classical application of ring closing metathesis to make microcycles with alkenes. So this is also something we could demonstrate using this carbon-sulfur bond metathesis. So we were able to synthesize a quite large variety of different microcycles using this chemistry. Here you see the sulfone as the product in this table. The reason is that it's just for purification purposes, having the oxidized product was a little bit easier, but the actual ring closing metathesis is the left part of this equation shown here. Sort of an extension of this metathesis chemistry, just to show you a little bit future direction before I conclude with the last short project, is that now we've been focusing those metathesis reaction on having the same bonds so far in the presentation, right? We had 2 times the carbon-sulfur bonds. But in principle, you could imagine being more creative with this chemistry and actually have 2 different types of bonds or 2 different types of functional groups. And you could still have a reversible aryl transfer or functional group transfer process that could be a synthetically interesting class of reaction. And so there are 2 specific reactions that we have developed so far in the group that I just mentioned here in passing to show you a little bit this sort of future direction of this chemistry. We could take, for example, the thioether and an aryl cyanide, and a single catalytic system, a nickel 0 species, is able to reversibly cleave both type of carbon functional group bonds and actually find a way to exchange the 2 substituents, the 2 functional groups, and then go back to do the reductive elimination. And that allows you to do this again reversible, so the transfer process between those 2 different classes of functional groups of aryl species. And you can also do this with acid chlorides and iodides. Again, very interesting or unusual type of reactions where now you take your aryl iodide, it gets converted to an acid chloride. while simultaneously you have the acid chloride getting converted to the aryl iodide. I mean, if you're more interested by those transformations, I encourage you to look at the corresponding publications. Of course, we also identify driving forces to sort of make them synthetically useful. And so, there are quite a few interesting strategies and mechanistic studies that are, involved in this if you're more interested by this topic. All right, in the last 5 minutes or so, I'd like to tell you a little bit about one of the last frontiers in terms of this reversible chemistry in my group. So now I just told you about exchanging or transferring two functional groups between two different substrates, two different starting materials. But we've also been interested recently in doing this in an intramolecular fashion to try to develop new interesting types of isomerization or transposition reactions that could be useful in particular for molecular editing. The idea or the type of application is very simple, is that if you would have a complex starting material, like let's say a complex bioactive molecule, could be a drug or a natural product, if you would be able to identify catalytic reactions where you can selectively just move one of those functional groups, for example, a carbonyl, from one position in that molecule to a different position in the molecule, that would be a very powerful tool to generate isomers of that bioactive starting material in a one-step fashion, versus normally having to go through, sort of de novo synthesis, multi-step synthesis, to generate each of those isomers for testing purposes. So, if we could develop as a dream family of catalytic reactions where each catalyst is able to only move one type of polar functional group in a bioactive molecule, would have almost unlimited access to sort of probe chemical space and identify interesting lead compounds. A very well-known reaction in terms of isomerization is alkene chain walking. This is a very well-known reaction. As I said, in transition metal, catalysis has been leveraged for a lot of interesting chemistry. And so our idea was, can we develop a reaction that could do something analogous, but now not transferring an alkene functionality along a hydrocarbon chain, but actually transfer an oxygen atom formerly along a carbon chain, to really be able to take carbonyls, which are very common functional groups in bioactive compounds, and generate those isomers using this type of transposition reaction. So how did we look or develop this transformation? This was inspired by a classical name reaction in organic chemistry that's not used a lot in synthesis. It's the Vilga-Rode-Kindler reaction. You take a carbonyl, for example, an acetophenone. In the presence of a sulfonic acid, S8 at high temperature, and morpholine as a solvent, you're able to generate thioamides as primary products of this transformation. If you look at it, what's interesting is probably not the thioamide. This is a quite, atypical functionality. What I think is more interesting is the fact that you have been able to move that sort of functionality by up to 5 or I think even 10 carbon units in the literature. That really hints at the fact that this chemistry, this cocktail of reagents, is somehow able to move functional groups along a carbon chain. There is very, very little literature about the possible mechanism of this reaction, but based on simple deuterium labeling experiments, people at least have been able to pinpoint a potential enamine isomerization mechanism as sort of key in enabling this transformation, right? Where you would be able to formally, if you look at this equation, transfer that carbon nitrogen from carbon 1 to carbon 2 to generate a new enamine isomer in this transformation. So our idea with Ben and Yannick was quite simple, was, okay, if we have a cyclic system where we do not have the ability to go all the way to a CH3 unit at the end and irreversibly oxidize to the thioamide and consume the sulfur and the morpholine stoichiometrically, we might be able to use just a catalytic amount of an amine and a catalytic amount of sulfur to be able to effectively do what we aim to do, to have this transposition of the ketone moiety just around the ring to generate different isomers and potentially generate very interesting carbonyl compounds that would be hard to access using other methods. This is shown here, very simple sort of test reaction we did to verify this. So indeed, having catalytic amount of pyrrolidine and catalytic amount of S8, we were able in this model reaction to show that no matter whether we start from this 1A compound, which I guess you could call the para isomer, or the meta isomer 1B, we're in both cases able to reach thermodynamic equilibrium, which is favoring 1B, the meta isomer. So showing that we can effectively do this isomerization reaction and that the catalytic system is active enough to reach the thermodynamic equilibrium of those two isomeric structures. You might be wondering why this ratio is not 1-to-1. Why is it this kind of 5 to 6-to-1 ratio in favor of 1B? We rationalize this based on very simple sort of conformational analysis, where we think the meta isomer has one less diaxial interaction with one of the CH group that points at the axial position, and that makes effectively the meta isomer be having a more stable structure in the ground state, and thus, of course, this is what's driving the selectivity towards formation of this isomer of the compound. And this seems to be confirmed by very simple DFT calculations that really supports that the meta isomer is the preferred one in terms of thermodynamic stability. What can we do with this chemistry in terms of applications? It's particularly interesting in the context of steroid chemistry, because you can now very effectively, very efficiently sort of generate those isomers where the carbonyl has moved by just one carbon. And in many, many cases, those species were not known or took really multi-step synthesis to access, which I think really highlights the potential of this chemistry to generate sort of new isomeric structures in this chemistry, this type of reaction. I mean, again, just to walk you through this, you start with the carbonyl, of course, at this position here where the blue dot is in the product. This is the product of the reaction. And you have been able to move this carbonyl moiety, this oxo moiety by one carbon unit. And as I mentioned, just one example to showcase a little bit, the advantage of doing this over de novo synthesis is shown here, where this compound, this steroid derivative, was generated in 8 steps previously in the literature. Our chemistry allowed us to generate this effectively in 1 step. Of course, it's not the highest yield. But as I said, this is again the message, right? Is that if you have those easily accessible bioactive steroid derivatives, you're probably very interested to do, this quick reaction, 1 step to generate the isomeric structure and see, when you test it, whether it has interesting biological activity. All right, I'm sorry that took a little bit longer. I think also because we had the interruption in the middle. So now I'll wrap up. I presented you a few different sort of recent directions in terms of the research in my group. You've seen this molecular editing sort of theme at the beginning of the presentation, starting with ways to just insert one nitrogen atom into sort of bioactive scaffolds. Another approach was skeletal metallation, so how you can replace one atom in a ring system with a transition metal, in that case nickel, and then use it to derive it to many interesting sort of derivatives. Then in the second part, I showed you a little bit this sort of completely different approach to methods development that rely on catalytic reversible reactions, really trying to transfer groups such as HCN or Cl2 that are challenging to use as reagents in organic synthesis, and also trying to develop new formal bone metathesis reaction. And again, right, really try to show how those can be useful in terms of interdisciplinary applications, right? I think they're a very interesting direction, I think, is this idea of growing polymers with those new reversible reactions and being able to degrade them at the end of their life cycle. And at the end, I concluded with this sort of merger of the two topics with this very intriguing sort of oxo-walk reaction to generate carbonyl isomers. And yeah, I think that's it for today. And thanks again for the invitation. Sorry it took a little bit longer, but I'm happy to take a few questions now. Thanks again for your attention.
Thank you very much for a very interesting lecture. We saw insertion, elimination, migration, and so on in your lecture. And we have some time for questions, please. Dear colleagues, do you have any to build about this impressive chemistry? Okay, I have several. First is a is a question about shuttle catalysis. So it's very clear and very effective on the slides. But what about real life, about byproducts, and how many byproducts do you usually have in these reactions?
This is an excellent question, Valentin. Of course, in practice, I mean, you know, for the reactions where typically we form, for example, a gas as a byproduct, this is obviously not a problem, right? Those are very convenient reactions to run, and it's actually quite easy to separate the products. I would say the cases where it becomes a little bit trickier is, for example, in those functional group metathesis reaction that I briefly mentioned here. So, you know, you can obviously imagine that in this case here, you know, both, you know, you're going to have 4 different species at the end of your reactions potentially. And obviously the polarity of some of those might be quite similar. So in certain of those transformations, then, you know, separation, purification of the products can be a challenge. But this is really the case only for this type of transformations. In the first few examples I showed with like HCN transfer, it's actually fairly convenient because you lose, right, the isobutene as a byproduct. So that makes the reaction overall be very clean. Yeah. But no, but this is a great point, and this can be a concern.
Okay, thank you. Another question about your first part of lecture, nitrogen insertion. Does it work only with indole derivatives or also with other heterocycles?
So we definitely think it has to be an electron-rich an heterocycle. So we had some success. I didn't have a chance to put the reference today, but we have a follow-up article in Inorganic Letters where we're also able to get pyrroles to react a little bit. But in terms of substrate scope, it was much more limited. So if I recall correctly, we needed to have you know, certain substituents on the pyrrole scaffold to sort of get the desired reactivity. So much more limited. We've tried also a little bit to do, for example, porphyrins or, you know, other types of very interesting scaffolds, because if you could make the, you know, ring expanded products with those structures, it would be very interesting for material science. I don't think in those cases we had any reactivity, at least so far. So in summary, I'd say that the indole scaffold and related scaffolds, such as aza indoles, so, you know, if you have 1 nitrogen in the 6-member ring, those tend to be quite good substrates for the reaction. But beyond this, it's still a little bit challenging.
Yeah. Okay. And maybe last question about Lindan conversion. So you convert one toxic compound to benzene, another toxic compound. What about further use of benzene? And did you apply this this procedure in the real industry?
I mean, that's something where we're still looking at. I mean, I cannot really comment on the specific details of collaborations, but no, you obviously have a point that, you know, you generate benzene, but, you know, I think if this would be part of a continuous process, you know, I think people, of course, in industry know how to cleanly collect and use the benzene that you generate as a byproduct. So I don't think that's too much of a concern for them, but obviously it would be much nicer if, for example, we could develop a reaction where we could, let's say, generate cyclohexane, right, as the byproduct. That would be something that would address your concern more directly. And, this is something that has been on our to-do list as well to sort of, improve this type of reactions. Yeah. Yep.
Okay. Thank you very much. It's very impressive, and I obtained a lot of pleasure from your lecture. Dear colleagues, maybe you have other question? No. Okay. Thank you. Have a nice evening today, and maybe we will meet in Kharkiv after the war.
I hope so. Thanks a lot again for the invitation, Valentin. Have a
Thank
nice day.